Kobayashi from Tokyo University. And
thank you very much.
Will be somehow in between statistics and modeling. He will speak about estimating synaptic connectivity from parallel spike trains.
So I will share
your screen.
Yes, screen. Yes. Okay.
Yes, perfect. We see it.
Thank you very much.
And we see your mouse if you want to use it.
Yes. Okay. Thank you very much.
Please.
Yes. Hello, everyone. Good afternoon and good evening. I'm Ryota Kobayashi. And first of all, I'd like to thank Professor Sachio Dorote for giving me the opportunity of this talk. And today, I first explain, this is outline, and first I explain now why we are interested in estimating synaptic connectivity. And then we introduce two methods for estimating the connectivity from spike data. And finally, we discuss how this method performs to artificial data set. Okay, so let's go. And so as you know, our brain is a network of neurons. That is, the brain consists of a huge number of neurons, and the neurons are interacted via synapses. And here we focused on an important research question, how these neurons are connected and how they are interacting during information processing. And before going to the question, we summarize the key players, neurons and synapses. Okay, and this is a schematic of the neuron. And as you know, neuron is a basic element in the brain. And the neuron receives fluctuating input from thousands of excitatory and inhibitory neurons. And also due to the input, the voltage, especially for the behaving, the neurons in the behaving animals, the voltage fluctuating, and they generate spikes. So roughly speaking, the neuron is a sort of the stochastic unit, and the neuron generates spikes when the voltage reaches a threshold. And this figure shows how the neurons interact via synapses. And an excitatory neuron, shown in this magenta, transmits a positive current that pushes the voltage of the postsynaptic, this neuron. And when presynaptic neuron, yes. And similarly, if the neuron is inhibitory, they generate a negative current which inhibits the neuron. So the neuron interact via voltage, not spikes. So in this talk we focused on spikes. So the important things is the interaction is somehow stochastic if we have only spike data. So it doesn't affect spike directly. Okay. And one important information is the one good news is there is a greater advances in decoding neurons. And the number of neurons which we can decode simultaneously, and number of the recording duration are also continuously increasing. And this is a log plot about the papers which records multiple neurons. And the x-axis is a publication year, and the y-axis is how many neurons they collected simultaneously. And you can see nice log plot. And interestingly, there is exponential growth in the number of recorded neurons in every 7 years. And now recently we can record up to 10,000 of neurons. So this is this is a very good time to working for such a massively collected multiple spike trains. Yes, and this is a program we focus today. Let's assume we record spikes from a part of our brain, and it is shown in the colored neuron, magenta and cyan. And we recorded spike trains from these guys. And we want to infer the synaptic connectivity between the recorded neurons. And in the next section, I explain how to do it. Okay, so then we explain, we describe the methods for estimating synaptic connectivity from spike trains. And first of all, I explain the most standard method, the cross-correlation for estimating the synaptic connectivity from spike trains. Note that it was originally proposed in 60s, and it is much older than me. Yes, and this is a schema. This slide explained how to do it in cross-correlation. This method is a pairwise method. So if we collected hundreds of neurons, we always focus on each pair for estimating connections. So let's focus on a pair of neurons, which we call reference and target neuron. And R1, R2, R3, R4 represents the spike times of the reference neuron. And the T1, T2, T3, T4 represents those of the target neuron. And this method first calculates the cross-correlation data. This is first we focus on the first spike of the reference neuron and calculate the reference time, S, and we obtain the sort of the transformed spike train of the target. Then we move to second spike, and we calculate Tj minus R2, and we have a second and transformed spike. And by repeating this procedure for all the spikes of the reference neurons, we have a cross-correlation data. It is a target spike train shifted by each spike time of the reference, of the spike time of the reference neuron. And using this cross-correlation data, we construct a histogram of the, we counted the number of spikes of the cross-correlation data, and we construct a cross-correlation histogram. And how many neurons fires, for example, just after the spike of the reference neuron, and so on. And this is quite interpretable, and because the peak around 0 indicates the connection between these neurons, and the valley, if we found a dip here, it represents inhibitory interaction between the neurons. And if you found a peak in the right side of the plot, there is it indicates the connectivity from a reference to target. And if you find a peak in the left side, it indicates again the connection from target to reference. So it is quite easy to implement and easy to interpret. However, there is a problem, and this method is quite, as I explained, relatively simple and easy to implement. But this method and also applied to various data sets, and we found many interesting things. However, this method has a drawback. It is vulnerable to large fluctuations in neuronal activity, which can be problematic if we are interested in the spikes recorded from behaving animals like me. and this is an example recorded from a walking rat, CA1. And this detected peaks based on cross-correlation method. Significant peaks are shown in red circles. And you can find 2 peaks and 2 valleys. But we can see that this peak may be connection, but it's not clear whether this valleys and this peak is really synaptic connectivity or not. So to overcome this difficulty, of course, there are several attempts. One method is actually introduced in the previous talk with dithering. It is also called the jittering method. However, this method is not enough to solve this problem in animals, in the spike strains in the spiking data set. So we want to develop new methods for estimating synaptic connectivity. And this talk, I propose 2 extensions of the cross-correlation methods that overcomes the difficulty. It's a vulnerability to large fluctuating neuronal activity. So 2 methods is called glmcc and connect. First, I start from glmcc. And this method is a combination of generalized linear model of a point process and the cross-correlation method. And so the basic idea is shown in this figure. We first generate a cross-correlation data, but we didn't construct histograms. Instead of constructing a histogram, we fit generalized linear model, which considers the input from neighboring neurons, shown in green. So our glmcc method considers the external input from the neighbors. So, and this idea is shown in this slide. This idea is shown using equation, and we fit the cross-correlation data using this model, this type of point process model, and AS represents the input from other neurons. It can fluctuating freely, and J plus, this is a connection from a reference neuron to target neurons, and J minus, this is a connection from target to reference neurons, and the kappa is a exponential function with delay, so which models the synaptic interaction between 2 neurons. So by fitting this model, we can have the estimate how we can estimate the strong the connectivity between 2 neurons. And this is the result, and you can see this is a result applied to the CA1 data. And you can see this glmcc works nicely to this data set. And so for this procedure, we find the parameters AS, J plus, and J minus. And after finding parameters, we detect significant connections. This is done by likelihood ratio test. Simply, we calculate compare the likelihood, which we consider the connection, and we didn't consider the connection. So if we didn't consider the connection, so this model explains everything with input from the neighbor, something like that. And we follow the standard procedure for the likelihood ratio test. So this is the glmcc. This is a combination. That method is a combination of point process model and the cross correlation. And the next model, we explain the connect. This is a combination of the cross correlation and the neuronal networks. And we infer brain using neuronal networks. And this is this figure explained how to do it. And as we explained, we construct a cross-correlation histogram. And the key idea is we transform this cross-correlation histogram to image. Because so if so this histogram can be considered as a histogram, as an image of a black and white. So the higher count means more black, and the smaller counts means more white or something like that. So, and if we transform the images, we can use neuronal networks, which is now quite strong for analyzing image processing. So we applied the image processing neuronal network, and we trained the neuronal network whether this figure corresponds to the connected or not connected, something like that. And let's explain the result of these methods. And to validate this method, we simulate the population of neurons. And for this simulation, we simulated thousands of neurons. And and 800 is excitatory and 200 is inhibitory. And we connected random connection, and each PSP follows log normal and normal or something like that. It's something for it's considered to be physiologically relevant. And we obtained asynchronous irregular state, which is considered realistic. And from the simulation data set, we originally have thousands of neurons, and we assume we only recorded 20 neurons from the thousands of neurons. And from the spike train of 20 neurons, we tried to infer the connectivity, and this is the result. And since we simulated the network, so we have the true connectivity. And this matrix, and the true connectivity matrix is shown in this figure. And the magenta means a positive connection. And this area represents the strongness of the connection. So this connection shows a strong connection. And cyan means inhibitory connection. And again, the size of the square represents the amplitude of the connection. And you can see, and the bottom plots, we calculate the number of total errors in the 2020 neurons. So you can see while these previous methods relatively high number of errors, this glmcc is smallest. And you can see the jittering method is the method tends to be very severe to detect a connection. So the detected number of connectivity is very small compared to the true connectivity. And we also repeated this validation using various data sets. And you can see glmcc and connect. The performance is somehow comparable. This is simulation data from MAT model, and this is again Hojikin X-ray. And for MAT model, connect is slightly better. And for Hojikin X-ray model, glmcc is slightly better. But again, these two methods is much better than cross-correlation and the jittering method. Okay, finally, we want to explain, we want to show the web application for the experimentalists. And let's share another screen. Can you see?
Yes, now.
So this is a web application for our algorithm. And this is relatively simple, just uploading data and select data and connect and push the button. And this is a result of our web application. And again, this shows the connectivity, the magenta shows the positive and the cyan shows the negative. And the color, strong magenta shows a strong connection. And you can create a network if you have spike train data. And we can apply various methods, glmcc and connect and something like that. And you can also check the cross correlation using the web application. So please, if you are interested, you can play with it. So I now sent the information to chat. And the Python application is also applicable. So if you are interested, you can play. So let's return to my presentation. And so as I explained, we developed this method. And finally, before going to the conclusion, we want to discuss the pros and the cons of the proposed two methods. So the conclusion is that these two methods are somewhat complementary, because the glmcc method is based on cross-correlation method. So we can easily check the result. So the estimation result is somehow interpretable. But unfortunately, this method needs to choose some parameters for each dataset. It's somewhat difficult, and it can be somewhat tough for experimentalists. On the other side, the connect is easy to apply the dataset because it was already trained. So just to push the button. But unfortunately, the connect, everything works in the neuronal networks, and we don't know how it works. So the estimation result is not interpretable. So these are, I think these methods are complementary. Okay, sorry. So summary, this is a summary. Today we explain two methods, glmcc and connect, for estimating synaptic connectivity from spike trains. And these methods can estimate the connectivity more accurate than existing methods. And the web applications and the Python codes are ready for your analysis. And thanks for your attention. And finally, I would like to thank for all the collaborators here. Thank you very much.
Thank you. I see some questions.
Yes.
So the first is from Ost. I was wondering whether the methods you presented can be applied for partially observed network of neurons. Could you comment about this?
Yes, as I explained, maybe my explanation is not good, but indeed, we investigate the partially observed scenario. So we simulated thousands of neurons, but we assume we have only 20 spike trains of 20 neurons. So I think this scenario is exactly the partially observed network. And even this situation, glmcc and the connect can estimate, of course, not perfectly, but they can estimate much more accurate than the existing methods. Thank you very much.
The next question is from Pascal Helson. I did not understand how you can infer inhibitory connections from cross-correlogram matrix. If I understood well, spikes from reference to target needed to cause spikes of the target cell in order to decide there is a connection from reference to target. In the case of inhibitory neurons, we do not expect such a causality. Am I wrong?
The inhibitory case, we also somehow, maybe there is some example. Inhibitory case, we somehow expect, this is an example. We can see small, some valley dip. So we assume inhibitory neurons decrease the number of spike count just after the spike. So in this sense, we assume causality also for the inhibitory neurons. But this method does, but our method might not work if our method doesn't work if these neurons doesn't fire at all. If zero spikes, no inhibition. But all the methods cannot estimate it. Thank you very much.
I see Alessandra has a question. Please, Alessandra.
Yes, I prefer to speak if possible. So I wonder if you are aware of a similar technique that was published in 2016 by the Scherberger lab in Germany. They use CCHs from actual experimental data. They evaluate them for significance, and then they basically construct a graph from them in order to study the topological properties of such graph. In that case, since they have floating electrodes, they cannot talk about a direct, let's say, connection between neurons. They call it functional network topology, but I think in design it's similar to yours, and I think this is really interesting. But my question is, because I tried last year with some colleagues of mine to actually apply their technique to our data across different electrodes, and what we observed is that we needed a lot of data to estimate such, for instance, dense cross correlations, and sometimes it is not possible because you have finite data and experiments, and I wonder if instead this would partially solve our problems, because we don't need, maybe we need less power for doing such a test, if you ever try to apply it on parallel spike trains that come from experimental recordings.
Yes. And at least we applied already. So already in this paper, for example, we applied data from rat CA1. And in that case, we applied the data from 90 minutes recording.
Okay.
It's a relatively lot, and what we checked, some strong connection can be extracted from 30 minutes.
Okay, that's really good. Thank you.
That's really and helpful. Also the second paper and this one connect paper, we applied 20 or 10 minutes recording from a monkey, and it is somehow they at least this method returns some answer. So please apply if you are interested.
Yes, thank you. Thank you so much.
Yes, and if you have some issues, please tell me. I'm happy and to work on that. Yes.
I see a last question from Dimitra Mutka. Have you compared how the performance of glmcc compares to plain GLM methods?
Yes. Yes. We also compare the GLMC. Originally, we worked on GLM methods, and we compare the performance, and we at least we found the GLMCC method is much better, and it's especially useful when the data has a large fluctuation, like experimental data. Thank you very much.
I think it's time Thank you again, Ryota, for this talk that moved a lot of questions.
Thank you very much.
And